what is the common factor, if there is at all? And, what we really, my slides aren't advancing. I only get 2 slides. Oh, there we go. Okay. So that although, there are a lot of distinct pathways, they do converge in the CNS involvement. So about 70% of the LDs have neuropathological involvement. And so that begins to really represent not only kind of a commonality as common as it can be, but a particular consideration in terms of biomarkers for screening, diagnosis, management, treatment, even and treatment response across the LDs. So what we started to do is think about, well, what then can we leverage or capitalize on from the CNS involvement to be able to visualize the progression, the response to treatment, and or safety efficacy. Because again, those unifying characteristics lead to things like neuroinflammation and all the different cellular mechanisms breaking down, and then cellular injury followed by tissue, and then subsequently organ injury. So starting at the brain, we looked particularly across the landscape, across 166 clinical trials that specifically had data that was applicable to CNS involvement. And white matter was obviously the characteristic in terms of visualizing what happens in the brain. So imaging in terms of white matter atrophy. Clearly, with white matter atrophy, how then that relates to clinical outcomes becomes a little more indiscreet. And so we know that white matter pathology disrupts the functioning of neural networks. So we're kind of back where we started from the molecular mechanisms. But how can we take what we visualize in the brain and apply it to, again, accelerating actionable solutions for drug development tools, and what are potential biomarkers? And so visualizing that through magnetic, basically through MRI, is a way that we can be able to start to see at least how the progression is, at least to identify those lesions in the brain that may have very, very close similarities across several of the LDs. Now, obviously 70 is not going to happen. Even 70% of 70 is not going to happen, but there will be subsets that have very similar progressive tracks and or response tracks in the imaging platform. So to that end, I went back to the clinical trials, went back to the literature, went back to looking for some of the unmet needs based on what was published. If we know it's published, we kind of know what's not published, which is a lot. And there's active clinical trials with longitudinal data. Again, with white matter pathology, we need longitudinal data, which obviously adds another layer of complexity into being able to capture data on ends of ones with different follow-up. But we have several active trials. We have several completed trials and a multitude of publications that specifically highlight their longitudinal data with imaging. And as the advancements with imaging the technology increases, the types of MRI and the analysis of the MRI is drastically improved. And so white matter is obviously something that's very important in terms of atrophy, but we know there's going to be atrophy. How then is that in the functional aspects of the brain that's more into the gray matter? So we started to look at the gray matter applications in imaging, obviously a lot less. The technology requires a lot more advancement, but we did identify longitudinal data source for 3 longitudinal studies for MRI. So why am I telling you this? Is because that begins to represent a biomarker that we could start to look at what are the applications across the LDs. Again, MRI for pediatrics in general. I did an MRI study of 4 to 6 year olds that were neurotypical and healthy relative. And it was difficult to get those, even with the MRI time down to 4 minutes. It was very difficult, obviously, when you add in the complexity of illness with these diseases, much more difficult. So what then can we use maybe for a complementary biomarker? And so we started to look at circulating biomarkers, the demyelination, the synaptic vesicles, all those things have a biomarker that potentially could be used. And so we started looking at neurofilament light chain, which is a measure of axonal injury. And NFL acts like a neurotransmitter. It increases with brain injury. And so that became another aspect. There is a lot of emerging interest in NFL in other disease states. It's starting to become more collected, even for retrospective analysis in the LDs. So we're trying to establish these working groups. Again, we're just launching to be fluidic and try to kind of reach a consensus so that we can move the hypothesis-guided physiological approach forward for the LDs. And so I'm going to skip over the just quickly what our goals are as we launch the consortium is to really aggregate that data. I've told you why we're collecting that data, but how then can we take that collected data and have actionable solutions? And one of the main functions of that to be able to aggregate that is to be able to put them in a place that's harmonized and available for consortium members and the community and stakeholders. And so we're trying to get those in a place. And with that, we're going to, I'm going to turn it over to how we can take that data and put it in a place that's manageable. Turn it over to Colin.
This is an exciting time. We have a really brief time for this conversation. Some of this we've spoken in other meetings, so I'll make it really quick and I'll.
It's that one, but it doesn't work sometimes. That's it. Wait, it's the one. Yeah, that one. There you go.
Oops. Okay. One of the things that we're, as CPATH, we have a group of core competencies that, really haven't been spoken to as much, of course, the biomarkers, our clinical outcome assessments, looking at modeling and simulation, and then, regulatory science support. What feeds these areas are data. And so obviously you've heard about RDCA DAP, which was generously supported by FDA, and it will be the repository for the rare disease data that's being discussed for this project. One of the things that's unique about RDCA DAP, and I don't think we can say this is enough, it is capable of ingesting a large amount of data types. I mean, the institutional memory about how to manage data in RDCA DAP, we have people consistently asking us how to do this in multiple rare diseases because in many instances they're standing up something new and we can provide that support. And so relevant to this project and other rare diseases, we've increased our capacity around imaging to help us with biomarker discussions around imaging for multiple rare diseases. And so this platform continues to evolve and innovate as we begin and we learn more about what the needs are for the rare disease community. I've listed there a whole bunch of different trial types. We try to create a diverse landscape of data when we attack any specific rare disease and allow it to, focus around developing solutions that will ultimately lead to advancing drug development. I just wanted to give everybody, for those, is the other advantage of this is it's open source. It's, we have the ability to share data and to stimulate research in the community. And so what we do is we take in de-identified data. You can request a login with and with submitting a scientific plan. It allows you to use this data in ways, that will either work in, as a prior for a drug study or for specific, scientific question that a researcher may have, or even nonprofits sometimes will use this because we have some tools there that help them with statistics about understanding their own diseases. And so it's really built with the context of, being able to use by multiple stakeholders. We're in conversations now with regulators so that they can better use it to generate high questions for their own internal uses that are doing their reviews. So this is the platform. Anybody in this room can request access to it. There's a ton of data that you can end up playing with. This just gives you, it's got built-in functions. I'm not going to go into this great detail. It's very user-friendly. If you don't know R, you can have cohort builder, which allows you to build your own sort of queries against the data and specific data sets that you've requested access to. And so all the way to, building advanced models, with R or other statistical tools. And this just gives an example of reports. I won't go into great detail of these. It gives you an example of what the interface looks like internally, just for sake of time. If people have questions in the break, I'm happy to discuss this further. But the point is that it's got a lot of functionality that allows someone who's not maybe deeply analytical all the way to highly analytical to meet their needs in looking at data. This just gives you examples from Cohort Builder. Those are ready generated reports that can run for you, so you don't have to be an expert in this. And we also have tutorials and things that are available to how to run and use RDCA DAP. This just gives everything from scatter plots and functional plots and raw data, so you can see and explore this on your own. Again, it's very user friendly. And if anyone who's used REDCap, for example, could intuitively use this. So the only other point I really wanted to bring about from this is, just because there's a broad group of stakeholders here, is that we spend a lot of time executing DCAs and at CPath. Having worked on the site side and worked with sponsors in my previous lives, DCAs are all one of those things like everybody grunts about, and it's not necessarily a priority. The beauty thing about the DCA at CPath is that we act as the data custodian, we're not the data owner, and that you can set the level of access that's available to the public. So, for example, even though we try to encourage everyone to openly share, sponsors, for example, sometimes will say, we don't feel quite ready to share our data publicly, but you can use this in the consortium level activities that you all do. And we're perfectly happy with that, because we want to advance drug development in this space. And so, we have a wide range of support for that. The only other thing that becomes a challenge, and this is speaking to investigators and sponsors, is that we come across is that really be important when you're considering your informed consent forms. Sometimes it's a misstep. People just sort of think, maybe I should think about data sharing. Make sure that you think about that when you're designing your informed consent, because it can ultimately be a significant barrier to future use of your data. And for rare disease, everybody in this room can appreciate how important single data points are, because you can commit huge sections of the population to a project where you can't use that anymore. And so, being able to do and share, that's really important. So I put that note out there, and going back and trying to reconsent, everybody knows what that's like if you've done PET studies. That's a legitimate barrier. That's all I wanted to say for the points of this. And I want to applaud. I've been going to Inc. for a really long time. It's nice to see the growth and diversity in the pediatric programs. It's exciting to see 2 new programs standing up, and just the additional expertise and thoughts and cross-pollinations of conversations. And I think, as we begin to look at these questions in both rare and neonates, and I think that the communities can learn from each other. And that's kind of one of the things that's helpful with RDCA DAP, is because since we see so many different data types from so many different rare diseases, there's so many embedded lessons learned, because it's some of the same questions, right? And that institutional memory that's going to be shared in this community, even outside of the data, I think is really important. So thank you. Come on down. I want to introduce Victoria Niklas from Oak Hill Bio Limited as her Chief Medical Officer. And welcome you, and thank you for your talk.
Great, thank you. Oh, hello, everybody. It's a great pleasure to be here. I actually had the distinct pleasure of following this the asset that I'm working on, which I'll mention today, from Takeda Pharmaceuticals when it was out licensed to Oak Hill Bio. And I actually couldn't see myself going forward in my career without making that change to allow this very important intervention to come to patients. But I want to, whoops, we seem to be missing. No. So these are my disclosures. Chief Medical Officer at Oak Hill Bio. OHB neonatology has the asset OHB 607, which is human recombinant insulin-like growth factor 1 with its main binding protein, which is a subsidiary of Oak Hill Bio, the company where I'm Chief Medical Officer. And then I also am obviously going to discuss medications and molecules that are not approved for use, And then I'm also a scientific advisor on a number of biologically based biotech companies. So this is what I'm going to do today. I'm going to very quickly, in 10 minutes, rapid fire, I'm going to highlight the unmet medical need, which I'm sure you're all very well aware of. I'm going to emphasize, why biologically based therapies may be very unique to counteract some of the complications of bronchopulmonary dysplasia. And then I'm going to look very briefly at the current landscape, which is really a handful. And then I'm going to bring some consideration to the value proposition that biologics may actually be adding in terms of therapy for BPD. But I want to stop for a moment and then ask you to remember a time with me. And I'm not going to give a beautiful presentation that David Attenborough might give as he remembers the times in the past. But I certainly remember a time when pulmonary surfactant was making the transition from pre-approval to approval through the clinical trials. Now, I was a pediatric intern. This was some 35 years ago. And I remember very distinctly the differences between infants that were delivered within the shadow of Children's Hospital of Los Angeles that were fortunate to receive pulmonary surfactant in and around the time of birth to those that were transferred in with complications having not received pulmonary surfactant. So the terrible air leak syndromes that those of us that have worked in the neonatal space are very well aware of. Now, naively, as an intern, I thought, well, this is easy street. We give the surfactant, the babies pink up, the ventilator settings wean, and all is good. Well, certainly we impacted that early survival, made huge differences there, but that wasn't the end of it all, because essentially what surfactant provided was a platform for survival. So now we've created an entire population that are developing diseases that are very unique to that prematurity. And certainly entire careers have been built on the diseases of particularly extremely premature infants. And so the survival has dramatically improved. Prenatal interventions in the mother, postnatal transition with surfactant, general ventilation strategies. And that's been a remarkable contribution to the care. But what's challenging are the health outcomes. In fact, bronchopulmonary dysplasia is one of the most common complications of prematurity. And depending how you look at it, those numbers are actually increasing. And this isn't just a short-term problem that you encounter in the neonatal intensive care unit, but it has long-term impact and matters significantly in terms of its impact on both society and on patients and their families. Now, the multifactorial nature of bronchopulmonary dysplasia has really been leveraged by those existing drugs, whether they're label expansion or primary development and proof, but it's also been significantly impacted by the technology. The 2 ventilators that I had to use as an intern resident and then fellow committed to neonatology is now vastly different and significantly improved. And so we understand very well the mechanics of lung physiology and the challenges that brings to infants with bronchopulmonary dysplasia. But there's a lot less known about the subtypes of the disease. So those infants, particularly those that are born extremely premature, which are going to have different outcomes than infants that are born within the extremely premature population, but of the higher gestational age subgroup. So how can we do better? Well, we need to move beyond those medicines that treat symptoms of disease and instead look at what are the things that are mechanistically important to that disease. And I believe that's where biologics fit in, because they actually could target the underlying mechanism, much the way alpha-1 antitrypsin deficiency exists. There may be deficiency syndromes that impact the lung and other organs of these infants. So this is a work from Michelle Walsh, and we're going to add biological therapies here. And this is where pulmonary surfactant started the story and really is the gold standard replacement therapy. And so natural substances and how they matter. Well, there are many biologically based therapies that are potentially applicable to the newborn period. But I'm going to show you here one which I think from natural history data has a very clear correlation. So these are IGF-1 levels, extremely premature premature infants born across those gestational ages. The red dots are IGF-1 levels in infants that are born at those gestational ages. And the gray dots are actually those levels that you find in utero for the corresponding gestational ages. So that is a deficiency syndrome. And we've subsequently shown with very detailed pharmacologic evaluations that, yes, this is a deficiency. It's been validated in a prenatal lamb model, as well as in a rat model of BPD. If you give IGF-1 and its binding protein, you reverse the disease and you can actually target the development of the lung. So it's stimulating development within the lung and the air spaces. Now, there are also other observations in extremely premature infants that suggest there are other places where you could impact biology. And one is if you look in tracheal aspirates or you look in histologic sections of the lung, you find that mesenchymal stromal cells are reduced in infants, and actually, particularly in those that have chronic lung disease, the function of those mesenchymal stromal cells is reduced, suggesting, at least at a hypothesis, that they too are deficient. So if you take all of this and you go to clinical trials registries, either in the US or in Europe, and you basically look at, well, what are those clinical trials that are out there? And if I were to show you the laundry list of things that need to be cleaned up on those websites, we'd be going through 5 to 6 slides. But if you look at those trials that are recruiting, not yet recruiting, or active, not recruiting, and BPD is the endpoint, what you find is this. And so you can see the top 3 are cultured mesenchymal stromal stem cells. And then the bottom one, exobiologics, is actually extracellular vesicles. And the 2 at the bottom may not meet the hardcore definition of biologics, but I think they're worth recognizing here. Airway Therapeutics has a recombinant surfactant protein D, and we have someone from Airway Therapeutics here in the audience. And then the last is, of course, human recombinant IGF-1. Those are both recombinant technologies. So what are our considerations here? Well, clinical studies with umbilical cord derived mesenchymal stem cells or extracellular vesicles, which I haven't really mentioned, the data have not been reassuring. In fact, it's one of the most common term when you look at the clinical trials registry overall. They show safety, i.e. the infants don't develop problems, but you don't find any efficacy signals. And some of those studies have up to 60 infants in them. So the question is how long do you continue those types of studies? It's probably the wrong stem cell population if you want to take a regenerative approach, or it's simply not going to work. I think the potential for recombinant strategies in the face of a deficiency, particularly a defined length of a deficiency, so you don't need a stem cell therapy or a gene therapy for life. You just need to fix the problem for that short period of duration. That's where the money is. And in fact, I applaud all those centers that do that sort of subtractive analysis to look for things that could potentially be important. Human IGF-1 happens to be one of those growth factors that's absolutely critical for organ development during the latter part of gestation. So I think moving this all forward, we need to manage the risk. And one of the things I've learned, and I'm going to conclude in just a few short minutes, one of the things I've learned is biological drug development is enormously expensive. And moving from Takeda to a small biotech requires, and I think it's been mentioned in the audience today, requires a lot of money. And when you pitch neonatal assets to venture capital, it's an area that they are not necessarily familiar with, particularly when you tell them you're targeting the extremely premature subset born between 23 and 28 weeks. They nearly put down the shades. But I think we've made a tremendous inroads in educating venture capital in this area. And one of their biggest points of sensitivity is, what does your approval path look like? Is this an approvable endpoint? And so not being able to stand very strongly on the podium to say, yes, there have been approvals for bronchopulmonary dysplasia. So there's no roadmap here and no prior approvals, although bronchopulmonary dysplasia is an orphan disease indication in pediatrics. How it all looks in the end is really uncertain. We all who are targeting drug development for bronchopulmonary dysplasia need to do this together. So cooperation and collaboration comes up. So there's a tremendous urgency here, not only for the over 80,000 patients that fall into the subset that are born in markets with access to neonatal intensive care units, but all of those that go unnoticed in that area. So lots of biological therapies, are likely to come, but we need to put a really strict lens on it because we want to focus where the potential for success is very high and critical. Now, one of the things I love about Proverbs is you can make it fit for whatever you're talking about. It's sort of like your horoscope. Oh yeah, that's me. That's my day. But, lighting one candle is certainly very, very important. But the great news here is I feel like there's a tremendous amount of light in this room. So I'll stop there. Sorry if I ran over. Thank you.
Could the panelists come up, please? Thank you, everyone, for staying to this session right before lunch. We appreciate that. What we're going to do to start this out is we're going to have everyone introduce themselves, and we'll start with Ebony and work this way.
Hello, my name is Ebony Ford. I am CEO and founder of Miracle Mamas. We are a nonprofit organization that helps parents and caretakers of the premature, medically fragile, and disabled. I am also a 2-time micro-preemie mom.
Hi, my name is Dima Martini Drew, and I work with Astellas. I have experience with lysosomal storage diseases for a very long time and with other inborn errors of metabolism, as I was working to launch and develop treatment for lysosomal storage disease with Genzyme. So I witnessed Gaucher disease being treated, which was the first lysosomal storage disease to be treated. And I think Pompe is the last of the lysosomal diseases and glycogen storage disease. So I'm very happy to be here and to working with all of you and within the different consortia.
My name is Gerri Baer. I am a neonatologist and a medical officer at the FDA. In my old hat, I was the lead neonatologist in the Office of Pediatric Therapeutics. But about a year ago, I transitioned over to the Center for Drugs, where I am a team leader for hepatology and nutrition products.
And I'm Victoria Niklas, and you know me.
Hi, my name is hi, my name is Christine Hon. I'm a clinical we were in the Division of Rare Diseases and Medical Genetic. The therapeutic area that I'm focused on, reviewing every day is lysosomal disease, specifically MPS II, but I do have some prior experience with other lysosomal disease such as hypophosphatase and keto and some others. Thank you.
Thank you all. I'm going to start with you, Dr. Niklas. You discussed the current landscape of biological therapeutics for BPD. Can you elaborate on any promising advancements or research that we should be aware of?
So I think some of the things to think about are advancements with regard to delivery technologies. So aerosolization of various drugs as well as biologics to the lung, which is the delivery service. But the other is leveraging what's been done in other mucosal systems, such as the intestine and the gut, which I have studied for some 30 years. So there is a product in development by Avilis Bio, which is actually a microbiome-related product where they hope to leverage commensal flora and their ability to secrete certain bioproducts that actually will improve dysbiosis in the lung, which has been shown as distinct from infection, which has been shown to correlate with infants with BPD. Obviously, you take your hat off and say, let's not give a commensal bacteria and then end up with a new type of pneumonia, but is leveraging the products and the ability of the microbiome to impact the function of the organ. So I think keeping an eye on that will be very exciting.
That's fascinating.
Ebony, this will be for you. We've heard a little bit, we've been talking about BPD for a while now. And I guess one of the things that we need to hear, is as far as the drug development perspective is we want to include patients and their families in the drug development process. I mean, can you give us a little bit of a hint or at least a peek about, what your life experience with BPD has been, and then, what your hopes are for new therapies as you're hearing these conversations?
Absolutely. So my daughter Rain was born at 26 weeks. She weighed 1 pound, 15 ounces. We spent 3 months in the NICU. And immediately she began to have a pneumothorax. She had 2 within a 3-day span. And that was our first indication that something was very wrong. From the ventilator to the oscillator, she went on to the bubble CPAP. And then every time we would make strides to wean her off of that, she would have a collapse or something would happen and put us back to where we started back on the ventilator. And so I heard the term BPD probably about 3 weeks into our NICU stay. And as the doctors began to explain it to me, I really did not understand at that time how much it would have an effect on our lives. But in the coming months, it would drive us to nearly get a trachea. I had to advocate I'll say sternly advocate against the trachea because I really felt and believed that she would overcome it. But it landed us on oxygen for 3 years. At the height of the COVID pandemic, she remained on it. She even contracted COVID, and that almost put us at a trach. But with the help of corticosteroids, which I have a love-hate relationship with, she was able to overcome along with her standard everyday medication. She is now a fairly healthy 5-year-old who is still on medications that we take every day so that she can live her rambunctious and energetic life. But BPD is still a very real part of our lives. And to have a cure, to have something that would better manage this disease would be miraculous, not just in my life, but for those of us who have been ravished by this disease. There are birthday parties, there are memories, there are trips, things that we had to sacrifice that we will never get back because of this disease and all of the things that it has caused her to miss because we've done our job well as parents. She doesn't know how much she's missed. We've always tailored things around her. We've never made her sit still. We've always advocated to her, but nothing can take back the moments where she was walking around with, a pulse ox of 73. And we had to explain to her, you can't run, you can't walk, you need to sit, or we can't go to Disney World because we can't get clearance to fly with this, or we don't know how you'll do so. Those cures will be there really are no words for what a cure or a better management of BPD will do for families like mine and those who I mentor who are currently being held back by this disease. So I encourage all of you who are on the front lines to keep going. We are rooting for you. Certainly our lives will be better because of it.
Thanks, Ebony. Dr. Baer, in your role at the FDA, dovetailing into that very nicely, what initiatives or regulatory pathways are in place to encourage some of the, as Ebony described, development and approval of therapies for exactly addressing some of those concerns?
Well, dovetailing on Ms. Ford, who I haven't met before, but I would like to have a conversation with. I think one of the most important ways to engage the FDA is through the patient-focused drug development initiatives. FDA has solicited some parent feedback and patient feedback about BPD, but I think we need more parents like Ms. Ford and other parents and patients. We even have had patients, who are now adults, who have lung disease related to preterm birth. I think bringing those voices to the agency for the staff to hear, I think, is one of the most important initiatives that I've seen in the 8 years or 7 years that I've been kind of grinding out, with this problem and trying to help move the needle a little bit. We all know what the challenges are. We need the right, molecular and pathophysiologic target. We need to find the right population for the potential intervention. We still don't have clear disease definitions. As Victoria said, we still don't have clinical endpoints that are necessarily agreed upon, but we are getting closer. And with that patient-focused drug development, really understanding what's important to families, I think we can get there. I will also say, initiative-wise, INC has already leveraged some of the real-world evidence initiatives in CDER's Office of Medical Policy, as was discussed before, building that tremendous database, and now trying to use the data that have been gathered to potentially, improve the ability to understand which patients would be able to benefit from these new interventions, when to target the interventions, and to identify some potential candidate endpoints. Some of the other initiatives at FDA that can be helpful, in CBER, there's an interact meeting process, which is a kind of early development. When you have some proof of concept data, you can come in and talk with the agency in CBER with some of these biologics and discuss sort of early feasibility and what to do next. So there's a couple of other areas that FDA has available, and I think someone mentioned the orphan designation. The Office of Orphan Products has natural history grants and clinical trial grants, and I know at least one of the clinical trial grants in the past 5 years or so has been for BPD. And then, of course, FDA has a very strong neonatology team that is available across the agency to help with any of these programs, in any of the centers. So FDA is really trying. We know this is a big problem. We really want to hear from patients and parents to understand what's clinically meaningful, because I know one of the biggest sticking points is what is that endpoint, and we're getting closer.
This question is going to be for Dima and then Victoria. Pharmaceutical companies in rare diseases and very young patients, almost are a bit of a mission-driven activity, right? I mean, you're struggling because you have to, convince people to work in a space that may be perceived as high risk and small numbers of patients and whatnot. One of the comments that I, wanted you to address is, what role do pharmaceuticals companies have to stimulate, research in this space, but also then you're also uniquely positioned, to work with partnerships like CPATH and the patient community. And I guess, what value do you have, in those opportunities and as companies and as individuals?
Thank you for your question. Can you hear me? Okay. I didn't hear 100% of the question, but most of it at least. So I guess I understand what the question more or less is. If you'd like to interrupt me, please do so if I go off course. So I guess Astellas specifically as a company, so we would like to use our discovery engine in the different primary focuses that we have. So we have multiple possibilities with gene therapy or with mitochondrial biologies or targeted protein degradation, and all of which will treat rare diseases and more specifically lysosomal storage diseases. So our biggest problem is that we identify a molecule, say that increases something or decreases it, etc., and then there isn't any knowledge about the disease. Sometimes the knowledge is there because there is a treatable treatment available for this disease, but most of the times there isn't a knowledge. So I guess, and the diseases change. For example, Gaucher, before there was enzyme replacement therapy, the phenotype is different from the patient who is on enzyme replacement therapy. And for that, the biomarker changes. Depending on the modality, different clinical outcome is needed. With enzyme replacement therapy, you would know that the substrate is now in the blood. But if you are going to clean the accumulation, what shall you measure? If you give a gene therapy, how can you assess the irreversible damage to manage the expectations of the therapy and hope for a prevention doing it? So a lot of things change. And if you work with multiple modalities in the same time, you're hoping to serve the patients and probably in the future even start treating before the onset of symptoms and cure. But for how long, this is unknown, and that requires a new clinical outcome assessment and a new biomarker to show as soon as possible there's any decline, to realize that there is a decline and you need another intervention. And this is really like reading a crystal ball, more or less. So this is why those clinical trials will have to be lengthier and mostly within the real-world evidence platforms. So I think also to learn about the diseases who don't have a treatment. No one of us can do it alone. And you will need all the disciplines of the patient care, from the families and the parents to the nurses and the nurse practitioners to the treating physician, etc. And you can look at electronic medical records and have a situational analysis and have an idea of where the gaps are. But you need to build those gaps before you start planning your clinical development program. And no one can do it alone. And the consortium for lysosomal storage diseases for LSDs is an opportunity that, if you think about it, when we were developing a treatment for Pompe, if this was there, I think the time would have been so much shorter.
Yeah.
And sorry, if I may also elaborate on the availability of resources from the FDA and other regulators, is also giving us a little bit more courage, because sometimes we need to do something that wasn't done before. So this relationship and as early as we identify a modality is really necessary, and we need to be better at that. Thank you.
Do you want to comment?
Yeah, good. So I'm going to answer the question, I think, a little differently. And I had the opportunity to interface with my particular pipeline at Takeda, which is obviously a large pharmaceutical company that had acquired Shire, a company very committed to rare diseases. And TAC 607, as it was called in those days, came in through that pipeline. Well, large pharma obviously prioritizes and deprioritizes all the time. And that does not mean lack of interest or failure to believe. And so, we were very fortunate with this molecule to actually have the backing of the company to find the right home for the molecule. And that and five other pipelines are what Oak Hill Bio is founded around. And the heart of what we need to do and continue to do is really it's tangibly more visible in a small company. Your every breath depends upon that success. So I think having the right home for a molecule is critically important. And I'll conclude there.
Thank you. Dr. Hon, as a representative from the FDA, could you share your perspective on the regulatory challenges that, kind of encompass what they both spoke about?
I talk about my experience in, the reviewing the submission for rare diseases, particularly lysosomal disease. I think so far we have approved maybe 10 or 11 or so of enzyme replacement therapy for lysosomal disease, as well as a couple of non-lysosomal disease. So far, we are able to find biologics that are useful and helpful to treat the peripheral manifestation of the disease. But then so far, like you mentioned already, the neurological manifestations and how to treat it remain one of the major concerns in some of these patients. So our goal is to try to, get a therapy as soon as possible in the patient population who need that the most, and especially they have high unmet medical need. So one of the way to get it is that we want to help, and we will work with the companies and then trying to design a well and adequate control trial to get data that are interpretable for us to determine the effectiveness of the drugs. Without good data, good quality data, it is really, difficult to do our job to figure out whether the drug is working. So one of the challenges that I would like to highlight here is the natural history of the disease. So far, I think, with all this, therapy that was approved for lysosomal disease or rare diseases, we still have a lot of uncertainty about the natural history of it, particularly in respect to the neurological symptom of this patient. So I think it is critical for us to, characterize, the natural history of the symptom. And that you mentioned before that you identify a couple of biomarkers that may be helpful in elucidate, or characterize the time course or natural history of neurological symptom. I think it is a good opportunity for us to, for different stakeholders, come together and analyze whatever data that we have and figure out what is the natural history, and then help us to determine what is the best, solutions in a clinical trial to obtain good quality data to help, us to get approval for therapy that is helpful for the patient.
Okay. I'm going to switch gears a little bit and probably go a little bit off script. So warn y'all, is that, I think we've heard through different conversation pieces in portions and innovation in the way we do research around, lysosomal storage diseases and BPD. I mean, that once could be said for many, rare and other pediatric disease states. One of the topics that I think that we've alluded to a lot is the impact of potential biomarkers and whatnot. And then I think the other thing that I know CPATH is involved with in the BPD space is, looking at progression modeling and whatnot. I guess, could some of the panelists, and I'll just open the floor, as a comment, what impact can you project or suggest that has for your disease state or for molecules that you're discovering and you're working through? Because, I don't want to ad lib, but, put words in people's mouths, but want to hear what, positive things or what hopes you have some of the tools that are being developed or currently and then in future tense.
I'll start basically to say that I think, and I haven't taken advantage of the disease progression model available. Our eyes have been focused on other things, but I think that's really critical for us. If we can identify, those contemporary patients to compare with what we're doing so that we can steer in the right direction. And particularly if we can leverage information with regard to different ethnic and racial groups. Because at the moment, our clinical study does not consider that as a variable. Although we look at it statistically, we don't consider that in the subjects that we enroll. And I think there are definitely disease-related outcomes that are tied to that, to sex, to racial groups. But disease progression and leveraging everything that INC has to offer is something definitely that will be top of mind for me.
Okay. So I was going to say, you mentioned biomarkers and you mentioned real world evidence. And I think two of the areas that are just hungry for these tools, if we can operationalize them, are, identifying and characterizing the different types of BPD and Dr. Ashner's endotypes from several years ago. It's not all one disease. That's part of the problem, is that it's a very heterogeneous issue. And so using real world data and potentially even biomarkers to really try to tease out, and there's been a number of good publications in the past few years, trying to really characterize the different types and targeting the drugs to those patients who with the relevant types. And then the other area that I think would be fantastic is some kind of use of the real world evidence or biomarkers for enrichment of studies, to really know who are the right patients to target, who are the ones who are most likely to develop BPD, because your patients under 26 weeks gestation, a substantial number of them are going to develop BPD, but the variation is tremendous. And so really trying to hone in on which patients would benefit, I think would be ideal.
I think, Dima, do you have anything?
I think the colleagues mentioned all great points. For me, on a personal level, not as only an employee and as a physician who worked with patients and saw some of them make it and some of them unfortunately not make it. I think we really need to use the ability to accelerate. We even need to have a clinical trial to select the indication, or not necessarily clinical trial, but maybe real world evidence to select the indication to accelerate the first approval, but have a plan for the biggest number of population that can benefit from it, and not just say it's done and wait 20 years for real world evidence to expand the label, like colleagues mentioned earlier today.
I echo the response saying that, biomarker can be very helpful, in help us, identifying a subpopulation that will respond to the therapy. But biomarker had, a lot of different uses, not just, so it could be diagnostic monitoring. So, but I think the key is that for us to understand the natural history of the biomarker, does it correlate with the progression of the disease? And at what point that is the higher point we want to detect it? And after therapy, when it will drop, and then tell us that it is working. So I think it's all tied in, what we know, and then we are trying to figure out all this relationship. I think your platform, that if people can contribute to the data, will be very helpful in elucidate all these, factors.
Yeah, as we conclude the discussion, what do you think are, the next steps in terms of potentially how stakeholders can really be influencers in guiding, some of the challenges and addressing some of the challenges, and, collectively pushing through to some of those challenges? And Ebony, I'm interested in from a patient perspective, What are your thoughts in terms of that? And I want to ask all the panelists, but I really, from you, what would you think would be the next steps, and what we could all do as a collective, collaborative stakeholder voice?
I think that using all of your collective experience, clinical experience and what you have seen, and putting real life data. Numbers are awesome, but real lives can make an even greater impact. And so putting some of those numbers, bringing some of those families perhaps in front of these organizations, hearing stories like mine, and even unfortunately of some of those neonates who did not make it, putting those families in front to drive the inspiration, perhaps using those families to bring influence, campaigns, even if necessary. Social media is the way of the world, right? Using that tool that we use every day to bring that knowledge out in the forefront so that those who are in power, those who are in research will be further motivated to find the cure that is necessary.
Yeah. Okay. So I guess let's get on with it. is for me the most important thing, and that's great that the Lysosomal Disease Consortium is starting, and that we'll be able to work together to identify the gaps, but not focus on the gap as an obstacle, but rather identify the tools and the possibilities to kind of remove any hurdle behind acceleration and agility, and not wait. Every day is an irreversible damage that's happening to the patient that no drug will give back. So
I think leveraging patient and family experience is critical. I think on the part of the developers, folks who are developing the therapies, to really understand what's important to the families and make sure the trials, are feasible, and that they're, that what you're proposing is something that people would enroll in. And then again, I'll just put in a plug for the patient-focused drug development program at FDA. Really bringing that information right to the folks who are reviewing these products is really important.
Yeah, and I'll just echo what has been said before me, and certainly, leveraging the patient and the family is obviously very critical. And so, Gerri, your words are resonating in my brain, but as a physician, that's always been the guiding force for what I've done. So really bringing that to the industry, bringing it to my own company, and bringing it to the consortium, I think is where I stand as well. So I'll be there.
I agree with all the comments that were made before by all the speakers. So I think, one of the things I think is important is to be open mind and really listening to what other stakeholders are telling us. So that way we won't have miscommunication, because I think earlier we heard that there's a miscommunication between the company and FDA. So I think it's really important to be open mind, listen carefully, and all the stakeholders come together and identify a common goal, and then figure out some kind of solution to get to that goal.
Yeah, with that, I think that's really the collective of that is, what's really important. And I look forward as a scientific director for CPLD to work with, across the multitude of pediatric programs. But we're gonna end it there. I thank all the panelists. Dr. Baer said that we're hungry for data, but as a dietitian, I can tell you that there's no calories in data, but there is calories in the lunch out there. So go get some nourishment, and we'll be back in here.
Thank you.